So, hello again everyone, Microbehunter here, Oliver here, and welcome to another live stream, to another Saturday live stream. Today I would like to dedicate today's topic to onion cells. You see one here in the background, that's the root of an onion. And I'd like to talk a little bit about cell division, and I'd like to explain to you what cell division is, what mitosis is. It's a little bit of a biology lesson, really. I've been doing a lot of videos in the past about microscope hardware and also about different microscopy techniques. And I decided, well, today, why not place somewhat of an emphasis on a little bit biology, okay? So I hope it's also going to interest you. So today's video is going to be a little bit, I would say, less practical than my previous videos were, or my previous live streams were. But I hope that they're still interesting for you. I also want to make an important announcement. It's not really an announcement, just an important piece of information. I just added a link into the description to another video which I made about 2 years ago, where I actually show you the staining of onion cells so that you can make the chromosomes visible. So this video is, the link into the video is in the description, so maybe after the live stream or whenever you have time. And if you're still interested in this topic, then you're invited to watch that video, which is going to be much more practical in nature, where I actually show you how I'm staining the onion cells. Okay, and I have already received my first comments. Yep, we're looking forward to a live stream, and biology is what we like to see through a microscope after all. Yes, I need, before I start, I want to tell you something very briefly, and that's the following. You see, microscopy is, I'm going to take my glasses off. Microscopy is, of course, a nice hobby, but it does have occasionally one slight disadvantage, and that is that sometimes when you buy prepared microscope slides, like the one that I have here in the back, it can be a little bit abstract, and sometimes it is good to know a little bit of biological background in order to understand what you're seeing. And so for this reason, I decided that, well, I'm just going to do a little video about the stages of mitosis, and we're going to cell division, and we're going to look at the onion cells here in the back, and we're going to try to identify those stages. So what I'm going to do now is I have to split up this live stream a little bit into separate parts. So for the first couple of minutes, I'm going to do a little bit of theory, okay? So I'm going to play teacher. I hope I'm not going to scare too many people away, but I'm going to simply, for those of you who don't know so much about cell division and mitosis, I would like to simply give you some theoretical background, and then we're going to dive in, and we're going to have a closer look at the picture here in the back. If you have any questions about anything or any comments, please do feel free to comment in the chat, also in the comments section. And usually what happens is that it takes usually several hours to a day for after the live stream for the comments also to appear. So if you're a little bit late to the live stream, and if you cannot see and read the comments, then you have to wait a little bit, and after some time they will appear, and then you can watch the video. So what I'm going to do now is I'm going to play teacher now, and I'm going to show you here a couple of pictures that I've found online, because understanding this is a little bit important, so that we also understand the thing that we see through the microscope. So I got this from Wikipedia, both of them. And I would like to, so these are onion cells, drawings, and this is basically cell division, how it happens. And I would like to also explain a little bit of why cell division, what's actually going on here. So what we have is this is the so-called the cell cycle, and the cell cycle, that's kind of the life cycle of a cell. And we've got here one cell, and then what happens is over here you see that it starts to divide, and then you have two cells, and the whole cycle starts again. Okay, but what is really important is what actually happens inside here, because one of the important things that we should never forget is that, you see, living things have to pass on information to the next generation. So this sounds abstract, but I mean, this cell here, when it divides, and then when you have two cells over here, those two cells should be the same, but they should be also the same as the original cell. This kind of I mean, it's kind of obvious, yeah. If you have a but the question is how is this ensured? Or for example, another thing that is really you've never asked yourself this question, but I mean, I don't know. Let's take two animals, two dogs. Let's say two dogs, a male and a female dog, and they have baby dogs, baby pups, and the baby dogs are also dogs, obviously, and not cats. I mean, we all know that, but the question is, why is that? I mean, this is the question most people don't ask. How can it be that if two organisms, when they reproduce and make babies, offspring, how come the babies are of the same species as the parents? It's so obvious, and we know this so much, but we've never been really thinking about this. Or when a cell divides, how come both cells are the same? So what we need is we somehow need to have a system, or cells or organisms, they somehow need a system to pass on information to the next generation. And the system that they're using, that is of course the DNA. So what a cell has to do in order to ensure that those two cells are the same as the original cell, the cell has to ensure that both of these cells have the same DNA in them. And this is what the whole point process is about. And this is the difficult part really, is how can you make sure that both cells contain the same DNA. And what has to happen is the following. Here, this is the original cell, the parent cell, and then the DNA has to copy. It's being copied. So there's a cellular mechanism, enzymes are involved, and the DNA is copied. And then what has to happen is that the DNA has to be separated so that each cell receives exactly one copy. And this is the difficult part because in reality there is not only one copy of a DNA here, but there are many pieces of DNA in one cell. And when they're copied, then you have to make sure that each one of the daughter cells, you call them daughter cells, receives an identical copy. And that is a little bit the difficult thing. And this is what the process of mitosis is all about, and what I would like to explain to you, and where we can actually also see it under the microscope. So what we have here, for example, is supposed to be DNA. That's DNA. And what you see in here, and that's a little bit difficult here now, these are chromosomes. And these chromosomes, this is DNA. And it's very compact and very dense DNA. And what I've done here is the following. I've prepared actually some strings here. These are supposed to represent my chromosomes. Okay, and what we have is we have in a cell, we have originally a copy here of the DNA, and then the DNA is copied, and then it looks like this. Okay, and the two copies are held together here in the center. We call this the centromere, and this here is a chromosome. And then we have, for example, human beings have 46 of them in one cell. I'm just going to make two over here. And if you now take the DNA of one cell alone, of one, let's say, skin cell, and if you extend it and if you kind of pull it apart, it's about 2 meters long in one cell. That's a long DNA. And what you have to do now, or what the cell has to do when it divides, it has to make sure that each cell, each daughter cell, will receive exactly the same copies. And that's the difficult thing. So I'm going to demonstrate the steps over here. First using my strings here. Then I'm going to show them to you over here, and then we're going to have a look and we're going to try to find them under the microscope as well. Okay. So basically, what happens is that before the DNA has been copied and before the cell now divides, what happens is that those chromosomes start to form, and they are now inside the cell. Okay, and the identical copies are held together here at the centromere. And what happens next is that those chromosomes are pulled to the middle of the cell. Okay, so you've got a lot of them aligning up here in the, you call it the equator of the cell. But what happens now is the following, because they were not in the equator in the center of the cell yet, so they're pulled to the center. Okay, they're so-called spindle fibers. These are protein fibers that pull them to the center. And what happens then is when they're pulled to the center is that those arms of the chromosomes, you call them the arms of the chromosomes, they kind of fold backwards. Okay, so they're being pulled to the center, and and this one over here on the side as well, it's being pulled to the center. And as it's being pulled to the center, I have a problem right now doing this, but those arms fold backwards. And then what you have at the end is you have a lot of these chromosomes lining up here in the middle, and you see that the arms here are pointing outwards to the so-called to the equator of the cell. Yeah, so basically under the microscope, we won't see it lined up like this, but actually rather like this. Okay, and with the different parts of the chromosomes, arms, pointing outwards. Yeah? And then the reason why the cell does that is to create a symmetry. Okay, we want to make sure that the left and the right cell, the two daughter cells, that they get the same DNA. And at the beginning, before the cell divides, the DNA is kind of spread up all over the cell, and the cell wants to now create a symmetry. And for this reason, it's pulled to the center. And this, as this happens, those arms fold backwards. Okay, because after all, cell is mostly liquid, and then, so that's how it looks like. And then this is referred to as metaphase, where they line up in the middle. And then the following happens. This centromere, where the two identical DNA pieces, you call them the sister chromatids. Scientists always like to invent new terms. Yeah, they separate, and then the individual pieces of DNA are pulled apart. So I'm going to do it just like this here. Yeah. Okay. Yeah. So basically, what happens is that, okay. The identical pieces of DNA here, the sister chromatids, they separate. They're pulled apart. Okay, and as they're being pulled apart, again, the arms, they fold backwards like this. And this doesn't only happen once, but of course also happens with all of the other chromosomes. It's pulled apart. And they're pulled to the poles of the cell. And then what we see at the end, we see over here, that then here on one end it looks like this, and on the other end, on the other pole of the cell, it looks like this. So you see that the arms of the chromosomes are now pointing to the center. Okay. So this is basically then also something that you're able to see under the microscope. And then after that has happened, we have now separated the identical DNA. And then what will happen is that both of these chromosomes, they will disappear again. They will form loose DNA again, and then the cell is able to divide. And then you have one cell here on the left and one cell here on the right, and the whole cycle starts again. So this is basically, these are all of the stages here. So I just want to show you over here. Again, at the beginning, you have prophase, where the chromosome starts to appear. The metaphase, where they start to line up in the center. Then the next one is where they're separating. This is called anaphase. And then when you have the DNA separated, and when the cell actually starts to divide, this is called then telophase. And all of these stages and phases are actually here on this drawing. And one of the reasons why I like this drawing or why I like drawings in general is because everything is so much more clear over here. Under the microscope, you're going to see that it's much more blurry. But then again, it's sometimes surprising how many details you are able to see after all. So for example, these here, that's a prophase cell, prophase. Okay, you see that the chromosomes are starting to form. Then where is a metaphase? I don't know if there's a good metaphase diagram here. Actually, not a very good one. Maybe this one could be seen as a little bit as metaphase. This one very clearly anaphase, where they are pulled apart to the poles. And I do not see a very good telophase one, where it has divided. I think that this drawing over here does not represent all of the drawings very well. So maybe this here. These two cells have just divided again, and have formed a new nucleus. So this is a little bit the schematic way of looking at things. And what I would like to do now is the following. I'm going to very quickly show you now in large pictures that I just made today, very large enlargements over here of the different phases. So you see over here, these are the onion cells. And the very dark, almost black dot in the center, that is the nucleus. And you see that the nucleus is in those boxes, and those boxes, that's actually the cell wall of the cell. Okay. Chromosomes are stored in the nucleus of the cell. Does the nucleus get destroyed? Well, that's a good one. Thank you very much. Basically, what happens is the following: that in the what you have, I'm going to use again, this one over here, this drawing over here. What you see over here is this is an interface cell, and this interface cell, that is basically when the cell is not dividing. So you see over here, this is a nucleus, and this dark dot that you see in here, that's the nucleolus. Okay, this is basically a region inside the nucleus where ribosomal RNA, so-called rRNA synthesis happens. That's not so important for our purposes, okay? But what happens next is that when the cell is now about to divide, and when over here chromosomes start to form, then the nucleus will start to dissolve, because the nucleus contains a so-called a nuclear membrane, and this nuclear membrane then disappears. So the comment over here, chromosomes are stored in the nucleus of the cell. Strictly speaking, if you're very, very narrow-minded about this, okay, then this is not correct, because if you have a nucleus, then you do not yet have chromosomes. Okay, not yet. So I know it's a little bit of a technical detail. So if you want to be exact about it, you have to say that in the nucleus of the DNA, loose DNA is stored. And loose DNA has a different name. We call it a chromatin. However, very often in everyday conversation, you just say that we humans have 46 chromosomes in our nucleus. Strictly speaking, this is not correct, because when there's a nucleus present, the chromosomes are not yet present. They will form when the nucleus starts to disintegrate, as the kind of becomes destroyed, the membrane dissolves, and the chromosomes start to appear. And you see over here in anaphase, there is no nucleus present anymore. Yeah, and then later on in telophase, here after the cell has divided physically, this is then when again the nuclei start to reappear. Okay, so yeah, I go back over here. These are interface cells, and you see the dark dot in the center. That is the nucleus, so that's full of DNA. These cells have been stained. Now, I did not do that. This is a commercial sample, a commercial slide. And it was also microtomed, cut very thin. And the stain that they used, of course, stained DNA. Yeah, and you see those boxes. Well, those boxes, that's actually the cell wall. And then what happens next is the following. Always look in the cells in the center. You kind of see that you see those little squiggly lines, sausages almost. Yeah, these are the chromosomes that now start to form. Yeah, and the nucleus is still a little bit present, because you can see that those chromosomes are still kind of collected together in the center of the cell. So this kind of means that there must be still a nucleus present, but it's starting to dissolve and fall apart. Yeah, and then the chromosomes are floating around more freely in the cytoplasm of the cell. Yeah. And the next one here is this is what I kind of showed you, and this is a little bit difficult to see, but maybe you want to look at the bottom picture on the right side. You see actually that there are unfortunately I cannot point to it, because you see my hand disappears, right? But this is actually what I tried to make it clear, where you can actually see that when the chromosomes align in the middle of the cell, that the arms of the chromosomes are kind of pointing outwards. Okay, yeah, and there are many of them lined up. Yeah? And these are typical metaphase. Because they're actually that's not correct. Both of them have to bend over to one side, and over here the other one as well. Yeah. Now you can't see either. Let's forget about it. Yeah, in any case, you get the idea there. The arms of the chromosomes are pointing towards the side. Okay, and you can see this. That's metaphase. I always remember metaphase with middle. Yeah? And anaphase here, this is now a little bit easier to hold. Yeah, when it's kind of pulled apart, and then the arms again fold backwards. Okay, yeah, like this, and then you can see that from the poles of the cell, the chromosome arms are pointing inwards. Yeah? You still see that there's actually only one cell, right? But what happened now is that the DNA, the chromosomes have separated. We still only have one cell, so we did not yet physically divide the cell. All we have done up to this point is separate the DNA inside the cell. Yeah? And then the telophase, that is basically then when the chromosomes yeah, they start to form loose DNA again, and then you also have again the two nuclei for each cell reappearing, and this is also the time when the cell physically, the cytoplasm of the cell, the liquid contents of the cell physically starts to divide, and then you have two nuclei again. And yeah, those nuclei that you see over here in the center, those two that you see there, or also in the top picture, they kind of look a little bit like prophase because you can still see the chromosomes there. Yeah, kind of in a little bit slightly disorderly manner, and that's correct in the sense that the telophase is the reversal of prophase, so the chromosome slowly starts to form loose DNA again. Yeah? And if you now think that everything like this is a little bit static, okay, I'm going to now show you an interesting comparison video over here, because what you see over here in telophase is that a new cell wall is forming between the cells, because you know, the cell wall of onion cells is kind of rigid, yeah, so it's not very flexible. But I'm going to show you a video of a ciliate. Okay, look at this here. They're also at the end of telophase already. I gotta. Yeah, floating around, and they are now dividing. So they have already, the nucleus has already separated, the DNA has separated, and now the cytoplasm, the cell actually is dividing. And then you're going to see that we have two ciliates. They're still connected, but after a couple of seconds, you're going to see that the division process is complete. And this here is, of course, an example which kind of shows that the cell is kind of folding inwards in the center. It's almost like a balloon being squeezed together in the center. Right? And the onion cells cannot do that because they have this strong cell wall on the outside, which is very inflexible. So they have to build a new cell membrane and cell wall from the inside. Yep, here we go. Okay, yeah, now they're separate, and the video starts to loop. Okay, I just wanted to show you this to you because this concept of mitosis, of chromosomes moving, is actually happening in all eukaryotes. Eukaryotes are those cells that have a nucleus, and in contrast you have the prokaryotes. The bacteria belong to the prokaryotes, and they have a much simpler way of dividing. Yeah. So, but now actually I want to go back again to the microscope view over here, and I'm going to now show you a little bit, give you a short overview of the microscope slide. And then we're going to zoom in, and we're going to try to find a few of those dividing cells. Okay, so I'm going to decrease the magnification a little bit. Again, this is luckily, not luckily, but I did not have to make this because this would have been a very ooh, look, it's very dusty, the slide. But actually, it's horribly dusty. Yeah, should have wiped it off a little bit. You know what? I'm going to try to wipe this a little bit. I don't know if this is going to help a lot, but this is almost embarrassing. So, look, I use those microfiber cloths, but sometimes the dust is not on the outside. Sometimes the dust is actually on the inside of the slide, if the company didn't make it very carefully. But I think it might be a little bit better now. It's a bit better. Okay, so what you see over here is on the top, right next, where is this, over here? Yeah, that's one onion root. The second one over here, third one over here, and fourth one over here. So there are 4 onion root tips here that have been cut. And you might wonder a little bit, first of all, why onion roots? Of all possible plant cells that there are, why exactly onion roots? Well, because onion roots, like many roots, they like to grow very quickly, and therefore there are many dividing cells there. So this is a specimen. The sample actually has been prepared by the company specifically for observing cell divisions, and so that is the reason why onion roots. And the second question that you might want to ask a little bit is, let's look at the top one. It kind of looks a little bit like, I don't know, like it's tapered off on both ends. Why is that? Well, because if you make a thin section, then the section was cut diagonally. Okay, so that's the reason a little bit. If you cut it diagonally, then it will appear, the two-dimensional section will appear to be tapered off. Okay, that's the only reason. And what we want to do is we want to go to the place of the onion root where there are a lot of dividing cells, and simply by looking, let's look at the second one from the bottom, this one over here. Okay, if you look here, then you're going to see that there's a part here which is pretty dark. Okay, and over here, towards the other end over here, it's not so dark. It's actually much brighter. And the reason is why it's so dark is of course because over there there are many nuclei. Yeah? So the density of the cells is quite high here because the cells are smaller and there's simply also more cells here. So if you want to find some dividing cells, this is the place here at the tip that we have to look at. Okay, I'm going to do something else. I'm going to start here at the end because I want to show you something that, yeah, might surprise you a little bit, but it kind of explains a little bit a few things as well on how microscopic specimens are made. Okay, I need to make it a little bit brighter now. Okay, I need to refocus. Okay. You see, of course, the dark nuclei. Okay, the dark dots, these are nuclei, that's fine. You might also be able to see that the cells are quite long and thin. They're kind of running, running where, like this. Yeah, so you see those lines. But if you look into the cells here, you're probably going to see that actually there are more cells in here than there are nuclei. Not every cell. It's kind of difficult to see the cell wall anyway, but not every cell over here has a nucleus. I mean, you could say, well, the cells are kind of long and thin, and therefore the density of nuclei is also less. But honestly, there seem to be some cells that don't have a nucleus, and the reason is because we're looking at a thin two-dimensional cross-section. So if you have, for example, a three-dimensional cell, I'm going to use my radio control for the lamp here as an example of a cell. If let's say the nucleus is in here, okay? Let's say that this is the nucleus, right? And I'm taking a cross section only from the top up to here. And I'm not seeing the bottom part. Yeah, I'm just looking at a thin section, yeah, which is, let's say, above or below the nucleus. Then I'm not seeing the nucleus. Yeah, so this does not mean that the cells don't have a nucleus. It actually means that, the section is so thin that I've removed or cut away the nucleus. Okay. Again, here, if this circle here, that's supposed to be the nucleus of the cell, and I'm just looking at a section that goes across like this, yeah, then I'm of course not able to see it. Yeah? And this is one of the things that when interpreting microscopic images, you have to be a little bit aware, because you might think, oh, I found a cell without a nucleus, but in reality, no, it does have a nucleus, but actually you don't see it because simply by the preparation of the slide, it kind of, it disappeared. So let's go back here to the tip. You see that the cells become smaller, okay, and also the density is much higher. Yeah, and here we are at the tip of the onion, okay, and it's kind of difficult to see now the individual chromosomes. So I'm going to put on my glasses again, because it's easier for me to see than the monitor, because of course I'm also looking at the monitor. So I'm going up with the magnification. I have to turn up the light intensity, maybe open the condenser a little bit, okay, and this is how it looks like. Okay. Okay. This is how it looks like. And I think I have to go up with the magnification yet more a little bit. So I'm using now 40 times. So 60 times is the maximum that I want to use today. Okay. I have to, go up a little bit. I'm just realizing that it's still a little bit dark, okay, but I think it'll be fine. And one of the things that, you know what, I think I'm going to do it like this. What about putting in the condenser to concentrate the light a little bit? Totally forgot about this. Oop, I lost the focus. Up, yes, much better. Okay, I think this is much better. And this is now 60 times. I just want to show you something else for those of you who are a little bit interested in microscopic technique. My 60 times objective has a so-called a correction collar. So this is something, it's a really nice thing. There is a ring here on my 60 times objective which I can turn. And look what happens now to the image quality. Okay, this is called a correction collar, which some objectives can have. and I adjust it so that I get the best sharpness. This is not focusing. This is not focusing, but this correction collar adjusts for the thickness of the cover glass. Not all cover glasses have the same thickness. There is a certain tolerance, and or maybe also a thickness in the mounting medium, for example. It also counts towards the cover glass, and by turning the correction collar of the microscope, I'm able to adjust it in such a way that yeah, I'm getting the best image, the most clear image. Okay. Yes, to compensate for different thicknesses of the cover glass. Okay, just a side issue, but maybe if you've actually seen already, I can already see two cells that are in metaphase. And again, over here, okay, again my finger starts to disappear. Yeah, I'm pointing now here, okay. You see there's this dark line here going down in the center, this dark line, this is the equator, and then you see chromosomes pointing out towards the side. Also the cell here on top, yeah? You see those faint lines pointing outwards, okay? These are the chromosomes. Still, these two cells here are metaphase cells, yeah? Yeah, so these basically, these two cells are metaphase cells, yeah? There's a question, is that a correction collar or color? No, collar. Collar just like the collar of a shirt. Yeah, because like the shirt collar goes around the neck, and that's why they call it a correction collar or correction ring. It is also sometimes referred to as that. Yeah, yeah. So this is yeah, found in some better objectives. I don't think that the correction collar makes a huge difference now, but it's a nice feature to have. Yeah, it's a nice feature to have, and again, not all companies make those. But it's only used for the higher aperture microscope objectives. Okay, so yeah, so that is but over here, yeah, these are the cells of metaphase. But let's actually we should step through the whole process, the whole stage of all of the stages, and you see that most of the cells that you have here, they have a pretty dark nucleus. Like I don't know, let's point to so that's an interphase cell. Okay, but look at the one right next to it over here. You see that there are some tiny lines in here. These are already chromosomes forming. So I assume that this one over here could be about to divide, and the chromosomes forming means that this cell is in prophase. Okay, so the DNA starts to condense, yeah, and starts to form chromosomes. But those chromosomes are still scattered around in the cell. They're not in any organ, they're just randomly there. And therefore, the cell has to pull them to the equator, to the center of the cell, to create a symmetry, just like I showed you over here. Okay? So this was at the top was probably, or maybe this one over here as well, could be a prophase cell. You see that it's not quite as dark as the other cells. And you can say that there are those tiny lines in there, which could be the chromosomes. Yeah? As a matter of fact, when you go through a slide like this, you're going to see that the majority of the cells are in interphase, and then quite a few of them are in prophase, or here at the top. This look this one here. Where is it? This one. You see this? Yeah, typical prophase. All of those tiny lines here are the chromosomes. Yeah. So. Yeah, so interphase, yeah, those with the dark purple ones, and prophase. Metaphase, you have those where the chromosomes, yeah, the arms have to point outwards. And then, I don't know, I found somewhere an anaphase. I'm seeing one, but that's not a very good one. Okay, that's not a very good one. Yeah, it is anaphase. Okay, do you see this here? I need to point to it. Okay. You see that they're all exactly at my fingertip. These are chromosomes pointing towards the center. And on the other side over here, there are also chromosomes pointing to the center. So this is basically when the chromosomes are separated, yeah, and they fold back like this. But honestly, I've seen much nicer pictures of this. And what I've been doing with my students, very nice prophase over here. You see the chromosomes? Yeah. And what I've been doing with my students is I made them count cells. So for example, they have to count 100 cells and then tick off how many of them are in prophase, metaphase, anaphase, and telophase. And the more that you find of a certain of a cell of the cells in a certain stage, the longer the phase. So you see that most cells here are in interphase, and this means that interphase is the longest stage or the longest phase of the cell cycle. Here, do you see this here? Which one is this here? Do you see this? Exactly this one over here. Into which direction are the chromosomes pointing? Yeah, they're on the outside, pointing inwards. Anaphase. Up. I bumped against. See, when this happens, you have to find it again. I bumped against my stage. Okay. Yeah. Again, yeah, I've been here already. Huh? So yeah. So I got a few more comments. The biology and supporting microscopy is very helpful. Thank you. How fast is mitosis? How much time does it take? That's a good one. I heard and read once, correct me on that, that the cell cycle of fast dividing eukaryotic cells is around 2 or 3 hours. Okay, so this is what I read somewhere. However, what happens in reality is the following: is that the cells might actually start dividing, and then yeah, they might actually stop somewhere in the cell cycle and wait until the conditions are right to divide. I want to, not a very satisfactory answer. So I want to show you the following. This video over here of the dividing cell, as a matter of fact, I followed this cell here ever since it started to fold inwards. Okay. It was actually one cell, and a larger cell, and I could just see that it was folding inwards a little bit. And then I think I observed it around an hour or so. And then I could see that it started to fold more and more inwards. So, but I would say that ciliates are probably quite fast dividing. Yeah? Bacteria, for example, they can divide once every 20 to 30 minutes. Yeah. So this is really it depends a lot on the cells, obviously, but the cell division depends. Here now it divided again. Cell division depends also a lot on how fast the DNA can be copied. Okay, I just want to tell you some additional information which I consider kind of fascinating. Scientists discovered that around 90% of our DNA that we have in our body is so-called junk DNA. Junk DNA means that if you look at the DNA sequence, it doesn't make sense. It's almost like if you have a book. If I give you a book, and for the book is, let's say, 200 pages, 300 pages long, and of the 300 pages, I don't know, 250 pages are just ABC, 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 ABC. I mean, then you know, come on, there is no information there, right? And it's a little bit also with the DNA of a human being, is that if you look at the DNA sequence, there are large sections on the DNA that don't make sense because they're just repetitive. Okay, for millions of bases, it's just repetitive, the information. And the scientists were kind of wondering why. What's the point? And there is now a hypothesis or a speculation which directly relates to cell division, which I heard many years ago at the university, which I consider very fascinating. And what they said is the following: if you have so much DNA, like I mentioned, 2 meters of DNA per cell, it takes the cell a long time to copy the DNA. And because the DNA has to be copied first before the cell can divide. Therefore, the long DNA slows down cell division because the cell cannot divide faster because it takes the cell so much time to copy the DNA. And this kind of slows down the cell division and because the cell division generally therefore is slower, it increases the lifespan of the organism. Okay, because if my cells were to divide too quickly, then my lifespan would be too short. Because of another reason, because every time when the DNA is copied, now I'm getting really into molecular biology. Every time when the DNA is copied for technical reasons and chemical reasons, the ends start to become shorter, and this is also related to the aging process. Yeah? And so this means that the cells that I have in my body are a maximum of around 50 generations old. So an average cell in my body has divided 50 times, and then I was a fertilized egg cell. So there is some kind of a limit on how often the cells can divide, and you kind of it's a built-in limit, and in order to kind of slow down the division process, you have a very long DNA. Okay, so this is kind of a I would say a little bit more on the technical side. Yeah. So let's continue to look around. I'm going down with the magnification again to 40 times because I get a better overview this way. And what time is it now? It's 39. Yeah, and because I want to show you a second slide as well, where you can actually see it much better. Let's go up here. Maybe let's try this one over here. Maybe I find a few more. You've probably already noticed that there are not so many cells there where you can see the chromosomes. Yeah, it was only a handful that I was able to show you. That's correct. Because mitosis is much shorter than it's only a short part of the cell cycle. Yeah, maybe it's just to get an overview here a little bit. I mean, I have seen again a few cells in prophase. Again, prophase cell up. Where's this? Here, this is again a prophase cell. This one here. Yeah. But maybe it's a little bit easier if I do go up with the magnification a little bit again. I'm seeing a metaphase cell. Oh, what a coincidence. This one over here, again metaphase. Yeah. 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 So, is it possible to see meiosis under a microscope? Yes, meiosis can also be seen. But again, you'd have to get the correct. You'd have to get a cross section of the testes, because this is where meiosis happens if you want to use animal cells, or the ovaries maybe. Yeah, so it should be possible as well. But because the chromosome movement looks similar. Okay, what stain was used? That's a good question. I don't know which stain was used because the slide does not say so. But if you want to make chromosomes visible yourself, in the video link that I posted in the description, there is a stain that's called carmino acetate. It's a powder, and I think it's also used as a food coloring, carmino acetate, and this is the stain that is, it's red. This one is more purplish, but carmino acetate is also a standard stain that's used to stain DNA. Methylene blue can also be used to stain DNA. Yeah. So there are a variety of stains possible. Okay, carmino acetate is the one. So if you want to make your own your own samples like this, then please watch my other video. Okay, that I posted the link which I made some time ago, where I actually show you how you do this. And in this case, you're not cutting it, but you're kind of squeezing. You're kind of squeezing the sample. Yeah. Also works. I was not quite so successful finding a lot of cells, but with my other method. But there were a few where I could see the chromosomes. Also down here, lots of metaphase ones here. See this one over here? This one, yeah. Again, the chromosomes that are pointing outwards to the equator. Okay. So what what people are sometimes doing is there is a mathematical formula called the mitotic index, and this is basically the proportion of how many percent, or what is the proportion of cells that actually divide. And cells that you find in cancers or tumors, tumors for example, they have a high mitotic index. It's rapidly growing, or the tips of roots and shoots of plants rapidly growing. Yeah. And therefore, the mitotic index, the proportion of dividing cells, is high. Look what I found here. Do you see those two? Those two over here? Okay. That is telophase. Okay. Yeah, I know that because it's the two nuclei look very similar, and you got to watch very carefully. But maybe you can see a faint line between the two nuclei, a very faint line. Okay, between the two nuclei. This is where a new cell wall is being formed. It's called a cell plate. Yeah, so because plant cells cannot fold inwards because they have the rigid cell wall, yet the cell has to build a new cell wall and cell membrane in the center. That's called a cell plate. So that is basically something that you're also barely able to see here. So these cells are in telophase. You can see that the nuclei are forming again. Yeah. So What time is it? Oh, it's 43. You know what I want to do now, because I've been talking about this quite long now. I want to show you actually another slide where you do not see as many, not quite as many dividing cells, but the chromosomes are much nicer. Okay, so that's the reason why I wanted to show it to you, because yeah, here with the onion cells, you see, you gotta have a little bit of practice in order to see those. And the chromosomes are not quite as evident sometimes, but I like this one over here. This is the cross section of the ovary of a plant. So yeah, basically where the seeds are being formed then. Look, I need to refocus this. Every slide has its own. Yeah, look here again. So you know what, I'm gonna go all the way down with the magnification so that you can get a nice overview, okay, of how this looks like. Yeah, so that's basically which plant? I don't know. A lily. Yeah, it's a lily flower. Yeah, and it's a cross section of, yeah, and yeah, basically what you see here, those things here in the center, I think these are going to be then these must be the egg cells of the plant, and this is basically which forms the seed. And all those tiny dots now, it's not 4 times, not 10 times, all of those tiny dots that you see, of course, are nuclei. And here you see it very clearly, much nicer than before, that not every cell seems to have a nucleus. And the reason, as I told you before, is as well because simply during the preparation of the slide, yeah, the nucleus was cut away because it's a thin cross section. Yeah. Yeah, so I don't know what stain they used here, but if you use carmino acetate, then it will look red like this. And this is now the time when we can actually look at some of those cells in the hope of finding some that divide. Okay? And I will tell you right away that there are not so many, but those that I found look actually quite nice. So let's have a look here. Let's make it a little bit brighter, and maybe if you are a careful observer, you might already see a cell in anaphase right in the middle. Yeah, again, I'm gonna move it forward. Do you see this here? Okay, yeah, diagonally, these are the chromosomes. So I have to go up with the magnification yet more so that you're able to see better. Right now on next to my eye, okay? Yeah. These are the chromosomes. I think they look much nicer here in this sample. Yeah, but there are not quite as many of them. And a little bit higher as well here. Yeah, you see? Also when I focus, yeah, lots of chromosomes. Yeah, they're pulled to the equator of the cell. We can see DNA. It's honestly, I love this because it's really unbelievable what microscopes are able to do here. How many objectives you have on your microscope? It seems that there is a lot. I have got a 4 times. Look, how many objectives? Okay, let's start. I have a 4 times objective, a 10 times, a 20 times, a 40 times, a 60 times, a 100 times oil immersion objective, and a 40 times phase contrast objective. I don't know how many I have. Okay, so that's phase contrast here. I don't think that phase contrast actually would look very nice here. But let's have a look. You're gonna open this up. This is not really a good phase contrast. This is how phase contrast looks like. You see, okay, not. Yeah, you have to get the right objectives for the right specimen. Yeah, this is too bright. Yeah, so basically the reason why I have so many objectives is because it helps me to choose. It's because I'm doing live streams and then YouTube videos. It helps me it gives me a little bit more flexibility without having to crop. Yeah, are the cell membranes and the membrane of the nucleus made of the same material? Yes, but if so, why does the membrane of the nucleus dissolve but not the cell membrane? That's a let's put it this way. They're made of, all biological membranes are made of phospholipids and proteins, but different membranes have different proteins in them and different phospholipids. Okay, so it's like if you say, is it made of the same thing? And I'd have to say, yeah, actually it belongs to the same class of substances, to the same family of substances, phospholipids and proteins, but there are many of them, and the specific composition is different. Okay, yeah, and the process of the plasma membrane, or not the plasma, the nuclear membrane dissolving and so on, that's actually also something that I have to admit I have not read into yet, but that's actually something I can imagine. This has to do something with the solubility. Maybe it could be, and I'm now purely hypothesizing here, is that somehow maybe in the cytoplasm, I don't know, certain substances are probably released that cause the phospholipids not to stick together anymore. Yeah, I would guess this is a hypothesis right now. Yeah, so the answer that you to your question is if it's made of the same material, the answer is yeah, generally yes, but then if you go into details, no. Yeah, because different biological membranes are made of different phospholipids and proteins, but the class or the category is the same. Yeah, so but actually I wanted to zoom in a little bit in the hope of finding something. Again, so I have to turn up the light again here. Yeah. One of the nice things about those, by the way, I can recommend this if you don't know what to observe under the microscope, and if you need some quick specimens to look at, or if you want to make some nice pictures of something, then I do recommend permanent slides as well, because they can be easily shown to other people. And they kind of look nice, especially plant cross sections, because of this irregular pattern. It's nice for photography. Yeah? I recently picked up a microscope for mycology. It also comes in handy for home canning and botulism. Yes. I need to maybe explain this a little bit. In the old days, I don't know. Yeah, what's the old days? People used a lot to make their own canned food. As a matter of fact, even my grandparents used to do that. And I once asked them, how do you make canned food? And what you have to do is you have to make sure that if you have food in a can, it must be inside absolutely free of bacteria. Yeah, because otherwise they start to grow. Now, how do you do that? How can you make sure that you cook the food and you put it into a can, and when you close the can and seal it off, how can you make sure that there are no bacteria in there? Because from the air, some dirt, dust, bacteria can fall in. I bumped the microphone. And then my grandparents actually told me that what you do is you put the raw meat into the can, you close the can, and you boil the whole can. Okay. The problem with that is that some people did not boil it hot enough, and some bacteria belonging to Clostridium botulinum basically started to grow in there, and they produced the botulism toxin. We now know the botulism toxin because it's used also as an anti, how do you call this? They inject them and you get the wrinkles away and so on in very, very low concentrations. But actually it's a very, very severe form of food poisoning. It's a very, very potent toxin. And now the comment here is the following. Well, what you could do is could you not check the food, the botulism, the food for botulism? And I want to say, be careful here. The problem is, generally by using microscopes, is that sometimes the concentration of bacteria is much too low. They're still there, but too low to be seen. So if I put some cooked food under the microscope, I'm going to see way more cells of the cooked food than I'm able to see bacteria. And so, but they might still be present there, and they might have already produced the toxins. So one has to be really careful. And this was actually in the 19th century and so on, actually a problem in medicine, because they wanted to take samples from a person, a biopsy, for example, take a tissue sample to check for bacteria. But they had problems actually separating the bacteria from the body cells, because the body cells covered up everything. So that's the reason why some of those doctors at that time, or biologists, developed specific staining techniques, like for example the Gram stain, which allowed the bacterial cells to be stained differently from the body cells so that you're able to see them better. Okay, and this is actually to overcome this problem of seeing cells, bacteria that are in quite low concentration. Yeah, so botulism for Botox. Yes, Botox is the short form for botulism toxin. Okay, same thing with a different. The Botox for this cosmetic thing is used in a much lower concentration, because what Botox does is it relaxes muscles. And when you now get the food poisoning, the Botox food poisoning, then you stop breathing because your muscles are not able to contract anymore. Okay, so that is the thing that I just wanted to clarify here a little bit. Yeah. So and even nowadays, botulism is a problem where you have to be careful when, for example, that's why you should not give honey or raw honey to babies, because it could be that there are some botulism bacteria in there. Yeah, and the immune system of the baby is not prepared yet for that. Yeah. Wow, I sidetracked a lot. I love doing this sidetracking. I actually wanted to talk about cell division, ended up with talking about botulism, but that's okay. That's why it's a live stream. Yeah, where I can interact with the questions of my viewers. Okay, yeah. So that is, maybe, I don't know if I'm able to find some more of these. Yeah, but yeah, I've seen some more here. Keep on sidetracking. It's interesting. You know what? I gotta be honest with you. Yes, I am also a high school teacher, and I love sidetracking during the classes. And one of my graduate students many years ago, when she said goodbye to me to go off to university, she said, one last comment, please do not stop sidetracking, because all of those stories, yeah, she also considered interesting. Look at this. Look, isn't this nice? Look, I'm going to point to this. This is a huge one. Look at all of those chromosomes here. Must be also metaphase. Yeah, all of those chromosomes. Yeah, and over here as well, metaphase again. Why so many metaphase? Yeah, metaphase here. Yeah, again, you see that the chromosome arms, they are pointing outwards. Yeah. Thinking about it, I actually might give my students a link to this video. They don't have to cover it during class. Cell division saves me some time. Yeah, this one over here on the side, probably prophase. Okay, because you see that it's not quite as dark anymore. Prophase cells, this nucleus is slowly going to start to dissolve, and it's not quite as dark. So you are able to see the structure. It's kind of a little bit granulated. Yeah, so these are the individual chromosomes that now start to form. Also over here, you see, I don't know what this could be, what stage this could be, maybe also prophase, but you see over here also chromosomes visible. Oh, I bumped again into my microphone. So it does require patience to see all these things. But the reason why I've been doing this, or one of the reasons, is not only because I wanted to talk about cell division, but simply also to kind of show you a little bit that sometimes when you look at a slide, you see those things, but you just overlook them because you don't know what you're supposed to look at. Okay? And then things don't make sense. And sometimes if you know a little bit of the biological background material, a biological background, then it becomes a little bit more interesting. Another side track. Okay, if you look here, for example, again, that's a plant cell. You see these are the cell walls, right? Okay, we see the nucleus because it has been stained. But the early microscopists essentially were immensely fascinated in the cell wall of plants, because that's the thing that they could see. They could not see all the inside. The microscopes wasn't good enough yet. Maybe they didn't have the staining available yet. So they were massively interested in the cell wall. And as a matter of fact, there was even a debate going on whether the cell wall is not the living part of a living thing. So maybe the inside is dead or empty. It's like a sponge, they thought. Yeah, and the cell wall, that's the living part. As a matter of fact, it's exactly the other way around. Yeah? But they could not see the center because their microscopes wasn't good enough, or they didn't have the staining techniques available. Yeah, and that's why the early microscopists or early, they placed a strong emphasis on plant cell walls. Yeah, and I actually read a report somewhere that there was actually some folks got an award for the solving of the mystery of the cell wall, because they somehow were able to prove that actually cells have a living contents, which is actually the thing that's alive. Yeah. Over here, also you can see that not all cells have an equally thick cell wall. So this one over here, it's much thicker, also because it's important for support. Yeah. Mitochondria have their own DNA. So I'm reading again off a comment here. Mitochondria have their own DNA or RNA. Well, both. They also make RNA, but they have their own DNA. They surely must divide somehow. They do. Is it in a different process? Has it been observed? Of course, it has been observed. And as a matter of fact, they divide like bacteria. By the way, just like chloroplasts, the green cell organelles in plants, they too have their own DNA. Okay, not only that, they have their own ribosomes. And what scientists discovered, and that's when I heard this the first time, I thought, what? Are you serious? No, but that is there is solid evidence for this. Is mitochondria and chloroplasts not only have their own DNA, they have their own ribosomes. They divide on their own because they once were separate cells that moved into a host cell. And you can basically, this is called endosymbiosis. This basically means that you have essentially the mitochondria, which are responsible for cell respiration. You call them the powerhouse of the cell because they provide energy for the cell. They basically, long time ago, were separate bacteria. Of course, the scientists made DNA studies, and of course they discovered the mitochondria, they come from the so-called the proteobacteria, the purple bacteria, and the chloroplasts are quite related to the cyanobacteria. Yeah, so that is called the endosymbiosis. And when I heard this, I thought, what is this crazy? What do you mean you have got cells living in cells? But that is something that you can observe almost on an everyday basis. Even I, some time ago, made a short video on my other channel where I could see a ciliate, a paramecium containing endosymbionts, photosynthetic bacteria in there who moved in. Yeah, so that is nothing unusual, really. And if you look in nature, it's quite common. There are even some algae out there, I couldn't believe this myself, where it is possible to remove the host cell and the chloroplasts are able to divide and live on their own. So the codependency has not established yet. Yeah. So that's quite interesting. So we are not a single species. Well, it's... Well, it depends how you define species. Obviously, per definition, we are a single species, but strictly speaking, correct. Going back in time, we are made of multiple cells and cells in cells that are now called chloroplasts. Yeah. So in that sense, it's interesting. A little bit is what is individuality in that sense? Yeah. It goes it's even worse. I mean. For example, bacteria are significantly smaller than our eukaryotic cells. And bacteria have 1/10 of a diameter of our body cells, approximately. 1/10 of a diameter means 1/1000th of the volume. And as a matter of fact, we have more bacteria growing on our body and in our digestive system than we have body cells. As a matter of fact, especially in our digestive system, we have, and on our skin. So strictly speaking, if you're very strict about this, we're made more of cells that are not part of our body, okay, than we have body cells, because we have more bacteria. I'm made from the number more of bacteria than I'm made of body cells. Yeah, the exact ratio of bacterial cells to body cells is a little bit up to debate. They used to say 10 to 15 times as many bacteria growing on and in our body than we have body cells. They have revised this number now recently. I read they said it's approximately the same amount. These are all estimates, but the number of bacteria growing on in our body is crazy, is high. Yeah, or in our mouth, for example. Yeah, and that is possible because bacteria are significantly smaller than our own body cells. Yeah, yeah. So it's quite fascinating. All of this, and also, for example, when you look at like dividing cells under the microscope. Yeah. Here again, some beautiful. Yeah. Chromosomes. It's interesting for me as a biologist and as a curious observer of nature, even though I studied this stuff, you know, and I've got a major in microbiology, a minor in biochemistry, it's still fascinating that all of those complex life processes, ultimately, when you break it down, are complex chemical reactions. Yeah, and still, even though we use chemistry to explain many of these processes of life, still it wouldn't be sufficient to say living things is just chemistry, you know? Because there's so many other factors that also play a role. It's really fascinating, yeah. How also basic physical processes manifest themselves in complex behavior. I don't know, you cannot say cell division is behavior really, but yeah. But all of this is controlled. For example, another thing, oh my gosh, I'm sidetracking again and then I'm going to stop. How does a cell even know when it should divide? Yeah, there's also some of the things that scientists were kind of wondering about. How does a cell know when it has to divide? Well, the cell knows that when it is large enough. But how does it measure its own size? How does a cell measure its own size? That it says, okay, it has reached a certain size, now it's time to divide. So how does a cell measure how big it is? And it does that by actually changing internal concentrations of certain substances. Okay? Yeah, so basically as the cell grows, there's more water in the cell, and this means that certain chemicals drop in concentration, because if the number of molecules is the same inside the cell, and then when the cell grows, okay, and there's a larger volume, then the concentration drops. And by measuring the concentration of certain substances, the cell knows how big it is. And if the concentration of certain substances drops below a critical level, then this initiates cell division. Okay? Very, very briefly put. Yeah, sorry, I just joined. Well, sorry, I'm going to stop soon because I've been already online for 1 hour, but you can watch the video, okay? This sample that we're looking at is now the cross-section of a lily, and all of those red dots that you see are nuclei, the nucleus, and I'm looking today at cell division. And some minutes ago I showed you the cells of onions, and here these are the cells of a lily, and occasionally we are able to see chromosomes here. And the chromosomes here in the lily are much nicer, but there are not as many of them. So I lost everything here right now, okay? So if you want to see the cell division a little bit, so you have to watch the video when it is after it finished, and YouTube will put it online, and then you can, catch up on all of the things that you missed. Yeah. So again, here again you see that some of the nuclei are not quite as dense, and this means that the cell is in prophase. If carmino acetate stains DNA, does it also make mitochondria and other DNA carrying organelles visible? I guess it should. I just wonder if the concentration of DNA in the mitochondria is high enough for you to see it. Okay. So that's a little bit the question that I have. Okay. So yeah, I bought a microscope. Your videos were a great help. Thank you. Well, thank you for the thanks. And again, I can highly recommend that. I know even if it costs a little bit of money, sometimes those prepared slides are also pretty nice, because sometimes you're able to see things that you're not able to easily prepare at home. Yeah. Normally, of course, we hobby microscopists, we take water samples, we see movement, and all these things. Obviously, it's very nice, of ciliates and worms and rotifers and amoeba and so on. But there are also some interesting things that can be seen simply on commercial permanent slides, because we don't have sometimes access to all of those specimens. Yeah. Folks, 1 hour, 8 minutes. I've been online now. I think it's time to stop again. Look, I know this video was a little bit different than my previous videos. My previous videos were more focused on microscope technology and methods and so on. Today I did a little bit more biology with a little bit of sidetracking. Yeah, if you enjoy this type of format, please write comments. Of course, I'll try to kind of keep the contents of these live streams a little bit diverse. So I'll do Q&A sessions again, like I did before. Maybe show you again some stuff under the microscope, like today. Maybe do some do-it-yourself type of things. For example, I've actually been also considering maybe to show you how to make microscope filters. So like for oblique illumination. So there are plenty of topics, but if you have any specific topics that you'd like me to talk about, then please post them somewhere. I read, even if I do not respond to all of the comments, I read all of them. Sometimes responding does take a bit of a time. Okay, but please post. I like the format very much. Me too. Well, thank you. Many thanks for your time. I like doing this stuff anyway, so and I hope that I'm able to inspire people a little bit, because I think biology should be fun. And I will tell you now a little secret. I, as a biology teacher who's been teaching biology now for many years in high school, middle and high school, I feel that sometimes a classroom setting does not allow me to make lessons the way that I want them to be. It's much easier for me to simply show you for 1 hour stuff under the microscope, to simply have a little bit of a talk like this. And this is sometimes not possible in a classroom setting. For this reason, I feel that those YouTube and live streams and the open possibilities that are really unimaginable to me up to a few years ago. Well, I'm going to leave it at that. Again, if you have any specific wishes for things that I should talk about, again, please leave some comments. If you joined in late, the video will be online, of course. I wanted to simply now wish you all the best. Happy microbe hunting as always. And see you around next time. See you next week. And if you're new to the channel, do consider subscribing. And I do also have a main Microbehunter channel where I also post videos and shorts. But it's not in this channel, but in my other one. I wish you all the best. Okay, see you around. Enjoy microscopy. Until next week. Bye bye.